Today we are going to see about chromosomal mutation. Introduction: chromosomal mutation, change in structure of chromosomes, change in number of chromosomes. Introduction: mutation is a sudden change of a gene or chromosome from one form to another. It produces an alteration in the character under its control. Dobzhansky stated that mutation is a mistake or misprint in cell division. The term mutation was introduced by De Vries. Genes are excretingly stable units. These are reproduced and copied exactly during meiosis or gamete formation. Nevertheless, mistakes occasionally occur during the copying or replication of genes during meiosis. These mistakes represent mutation. Once a mutation occurs, it is then reproduced and copied exactly during meiosis. Mutations are the basis of discontinuous variation in population. Mutations may occur at chromosomal level or gene level. Chromosomal mutation: the changes in the structure and number of chromosomes genome are called genome mutations. Since these mutations occur at the chromosomal level, they are also called chromosomal aberration. The genome is defined as the total genetic material contained within the chromosomes of an organism. Humans have 46 chromosomes. Our 46 chromosomes present are genome. Genome mutations occur at a frequency of 1 by 10,000 to 1 by 10 lakh. Genomic mutations takes place due to abnormalities in cell divisions, especially during gametogenetic meiosis. They may occur due to change in structure of chromosomes or due to change in the chromosome number. Any change in individual genes has been considered in genome mutations. Genomic mutations are analyzed with cytological investigations of cell. Genomic mutation is of two types, namely, first, change in the structure of chromosome, second, change in the number of chromosome. Change in structure of chromosomes. The chromosome contains DNA. The change in the structure of chromosome bring about change in the number and arrangement of genes. There are four types, namely, first, deletion, second, duplication, third, inversion, fourth, translocation. Deletion. Deletion is a chromosomal aberration where a segment of the chromosome is lost. Here some genes are lost. Deletion is of two types, namely terminal deletion and intercalary deletion. In terminal deletion, a terminal segment is lost. In intercalary deletion, an intermediate segment of the chromosome is lost. When deletion occurs in one member of a homologous chromosome, a deletion loop is produced in the normal homologous chromosome. The deletion loop is formed by the segment opposite to the deleted segment. It occurs during pairing in meiosis. In human babies, deletion of a segment of chromosome number 5 causes disease called Cre-Du-Chat syndrome. The baby cries like a cat. It is mentally retarded with small head. Duplication. Duplication is a chromosomal aberration where a segment is repeated. Hence, a set of genes is present in double doses. Duplication produces position effect, a phenotypic effect produced by change in position of a gene or a group of genes is called position effect. Inversion. Inversion is a chromosomal aberration where a segment of chromosome breaks and reunites in the reverse order. In inversion, there is no loss or gain of genes, but the genes are rearranged in the reverse order. Inversion is of two types, namely pericentric inversion and paracentric inversion. In pericentric inversion, the centrome is included in the inverted segment. In paracentric inversion, the centrome is not included in the inverted segment. The chromosome with the inverted segment produces an inversion loop. Inversion prevents crossing over. It brings position effect. Translocation. Translocation is a chromosomal aberration where no homologous chromosomes exchange segments. Translocation produces a cross-shaped structure during pairing. Translocation causes position effect. Translocation alerts the linkage groups. Aneuploidy. Aneuploidy is a chromosomal aberration where there is a gain or loss of one or more chromosomes in a set. Aneuploidy is caused by non-disjunction of chromosomes. It is of three types, namely monosomy, nullisomy, trisomy. Monosomy. Monosomy is a chromosomal aberration where one chromosome is lost from a pair. It is represented by 2n-1. The monosomic individual has one chromosome less from the normal number of chromosomes. A monosomic Drosophila has 8 111 chromosomes, a monosomic man has 46 145 chromosomes. In an aneuploidy, it is produced in women when an egg without in capital X chromosome fuses with the sperm containing and capital X chromosome. It causes a syndrome called Turner syndrome. Nullisomy. Nullisomy is a chromosomal aberration where both chromosomes of a pair are lost. It is represented by 2n-2. It is an aneuploidy. A nullisomy is produced by the fusion of gametes having one chromosome less. Nullisomic individuals cannot survive. Trisomy. Trisomy is a chromosomal aberration where one chromosome is added to a pair. It is represented by 2n plus 1. A trisomic individual has an additional chromosome from the normal number. Thus, a trisomic Drosophila has 8 plus 9 minus 1 chromosome. A trisomic man has 46 plus 1 minus 47 chromosomes. It is an aneuploidy. Trisomy is caused by non-disjunction. There are two types of trisomy, namely trisomy of autosomes and trisomy of sex chromosomes. Trisomy of autosome is due to the addition of one chromosome to any one homologous pair of autosome. When a chromosome is added to 21 pair of autosome, it is called trisomy 21. Trisomy 21 in man causes a syndrome called Down syndrome. A trisomic man has 47 chromosomes instead of 46. They are mentally retarded. They have broad face and flat stubby nose. Trisomy of sex chromosome is due to the addition of one sex chromosome. When an capital X chromosome is added to a man, he has 47 chromosomes, 22 capital A plus capital X capital X capital Y. It causes a syndrome called Klinefelter syndrome. Euploidy. Euploidy is a chromosomal aberration involving change in the number of chromosome sets. It is of two types, namely haploidy, polyploidy. Haploidy or monoploidy. The basic set of chromosomes in any species is haploid. Each chromosome is represented singly, that is capital N number. The gametes carry haploid number of chromosomes. During fertilization, the parental chromosome unite together by the fusion of gametes forming diploid number 2n of chromosomes. Sometimes in the life of an animal, a set of chromosomes will be lost and it leads to haploid. So, some characters which are present in the parent will be lost from the resulting individual. Polyploidy. Polyploidy is the condition in which an organism contains more than the usual two sets of chromosomes. Such animals are said to be polyploid. Polyploid organisms may have 3, 4 or more sets of chromosomes and they are called triploids 3n, tetraploids 4n, pentaploids 5n, hexaploids 6n, heptaploids 7n, octoploids rn, nanoploids 9n, decaploids 10n and so on. Polyploidy may be autoploidy or haloploidy. In autoploidy, the chromosome sets are derived from the same species and no addition of new genes occur. In the haloploidy, the chromosome sets are derived from the distinct species. It involves the addition of new genes, hence much variations occur in organisms. These variations are inherited by the offspring which seem to be different from their parents. Origin of polyploidy. The polyploidy condition arises owing to the abnormalities in cell division. During gamete formation, the homologous chromosomes may not separate completely. Hence, one cell is formed with diploid number of chromosomes and the other cell without chromosomes. During fertilization, such a gamete with two sets of chromosomes pair with the gamete having haploid n. Number of chromosomes resulting individual is a tetraploid 4n. When both diploid gametes belong to the same species, the resulting tetraploid is called autotetraploid. When the two diploid gametes belong to two different species, the resulting tetraploid is called allotetraploid. Thank you.